Welcome, everyone, to our closing keynote. I can't believe it's already here. Today's keynote is all about genetic therapies and how it will be a something for everyone. This closing keynote will look at all the possibilities and challenges of genetic therapies on the horizon. My name is Shelby Lubert, as you already know, and one of this year's co-chairs, along with Cindy George. I will be introducing our moderator for today in just a moment. Before we get started, I want to go over a few items related to the technology and how we can all interact. First, closed captioning is available in English. You can turn them on by moving your mouse over the video feed and clicking the CC icon that will pop up at the bottom of the screen. Under the Stage tab on the right side of your screen, you'll notice the chat for everyone who is in this keynote. If you are in the Event tab chat, that is for the full event and not specific to this discussion. So be sure you are in the correct chat to engage with others in this keynote. We will try to take audience questions at the end, but this will be a jam-packed session. We encourage you to submit your questions at any time in the Q&A box and not in the chat feed, please. We do not want to miss them, and there will be a potential that you could get your questions answered in a typed reply from Dr. J.P. Clancy that is helping out today. You can find the Q&A by making sure that you are in the Stage tab on the right side of your screen. Then select Q&A. In the Q&A box, you can also like questions submitted, which will help us prioritize based on popularity. Make sure to check back to those periodically to find replies. We won't be able to answer questions about individual diagnoses or treatment plans, and we encourage you to use the information that you learned today to have conversations with your care team. We will also have lots of resources connected to this keynote, so please make sure you download them in the reception area. If you are having any technology problems during the keynote, you can find the help desk by clicking the Expo icon on the left side of your screen. CF Foundation IT support are there to help if you are having any trouble during the event. Now, I'd like to introduce Nima Shandal, an adult living with CF. Her story is powerful, and I am so honored that we could feature her during this closeout of ResearchCon.
Thank you, Shelby. Hello, and again, my name is Nima, and I'm an adult living with cystic fibrosis. I'm 39 years old and wasn't diagnosed with CF until 10 years ago. Throughout my life, my CF was continuously overlooked despite my overt symptoms. It was because of my Asian ethnicity that CF wasn't considered as a possible diagnosis. So, it started with an X-ray. There I was, visiting a pulmonologist for the first time. The year was 2012, and I was 29 years old, and I had had a chronic productive cough for almost a year now. I felt fine otherwise. The pulmonologist suggested we start with an X-ray. I even asked him, could this possibly be cystic fibrosis, perhaps a lesser version of it? He said it was possible, but something we could look at further down the road. As a baby, my mom said that after I would sweat, I would have granules of salt on my head. Because of this, I ended up getting a sweat test. My dad distinctly remembers the person running the test saying to him, Asians don't get CF. My results put me in the gray zone, and they said I didn't have CF. At age 2, I was diagnosed with asthma, and throughout my childhood, I suffered from asthma attacks, and later on from bouts of bronchitis. Then, when in college, I started getting frequent sinus infections. I showed my medical records from the student health clinic to my pediatrician at home. He didn't believe I had chronic sinus infections because I had been diagnosed by a nurse practitioner and not a doctor. I was eventually referred to an allergist and then to an ear, nose, and throat doctor who performed 3 sinus surgeries over the course of 4 years. Afterwards, I still struggled with sinus infections, but to a lesser degree. So back to the pulmonologist. After the X-ray came a CT scan, followed by bronchoscopy, which cultured Pseudomonas aeruginosa, the organism virtually pathognomonic for cystic fibrosis. I was sent for a sweat test, which resulted as slightly positive, and I was referred to the local adult CF clinic, where I was officially diagnosed with cystic fibrosis. And what did I feel? Relief. Yeah, after 29 years of struggling, everything added up, and I finally had a name for my disease, a name and a treatment. The doctors at the clinic also told me that those gray zone results I had when I was tested as a baby would have diagnosed me with CF had I been born today and tested. When I told my ENT about my diagnosis, he said he could have kicked himself because he had let my ethnicity get in the way of him seeing the CF. I have 2 rare mutations, and because of my mutations, my diagnosis was delayed. Because of my mutations, I was not eligible for modulators. Because of my mutations, my CF progressed to the point of me needing a transplant. November 21st, 2018, 3.5 years ago, I received a double lung transplant. Had there been a modulator that my mutation responded to available, my transplant may have been delayed. Had that modulator been available, I may not have needed a transplant. It is clearly known that CF predominantly affects white non-Hispanic people, but it is a myth that only white people get CF. Those of Hispanic, Native American, African, and Asian heritage are all represented in the CF community. According to the CF Foundation 2020 patient registry, 3.5% of CF patients are Black, 0.5% Asian, and 0.3% Native American. 9.6% of CF patients identify as Hispanic. Sadly, people from these racial and ethnic minority groups are less likely to be eligible for modulator therapy based on their mutation. 10% of the CF community have a combination of 2 rare mutations, and a majority of them can't benefit from a modulator. This 10% represents an underserved demographic. This 10% needs alternative therapies to correct the underlying cause of their disease. Genetic-based therapies are on the horizon, and they are the alternative therapies being designed for that last 10% of the CF community. The slogan for the ResearchCon this year is, "Your story matters in research." Yes, your story matters and your mutation matters. Knowing your mutation is the first step in knowing what drugs you can take and what research trials you can participate in. I participate in most of the CF research studies I'm offered, even being post-transplant, because even if today's research may not benefit me now, it may benefit me or someone like me in the future. And I greatly encourage all of you to participate in research trials too, if able. With research focusing on rare mutations, our underserved demographic and the CF community is closer to having advanced therapeutics and are closer to the day when we have a cure. Now, I'd like to introduce our clinicians here today. First, you will hear a video presentation from Dr. Manu Jain, the director of the Adult CF program at Northwestern University and co-principal investigator of the Therapeutics Department Network Center at Northwestern. Then afterwards, you will hear live from Dr. Lynn Quatel, Vice Chair of the CF Foundation's Data Safety Monitoring Board.
Good afternoon, and thank you, Nima, for sharing your perspective on living with CF. Obviously, Trikafta has been a big step forward for many people with CF, and we are thankful, but the work is not done. Many people with CF are not eligible for Trikafta, which includes a disproportionately higher percentage of people of color, and they clearly have a need. So my goal today is to provide an update on the continuing efforts to find something for everyone. Towards that goal, I'm going to tell you about the genetic therapies on our horizon, both the possibilities and the challenges that lie ahead. So without further ado, this is a disclosure from CFF, and this is my disclosure. So before we get into the core of my presentation, I thought it would be helpful to step back for a second and think about what we may have learned in our biology class in high school with respect to the DNA and mRNA and protein. Remember, it's the DNA that represents a genetic code, and this is what is abnormal in CF when you have CFTR mutations. And it's the gene mutations that make abnormal mRNA in CF that lead to abnormal protein function. So conceptually, if you could correct either the gene, the mRNA, or the protein, you could target the underlying cause of CF. And so this is an important concept to remember as we talk about some of the genetic therapies in the rest of my presentation. So when we talk about CF2 mutations, it's important to remember that this is a very complicated set of mutations. When you look at the gene, there are two big components of the gene. They're made up of something called exons, which are sort of the part of the gene that make the mRNA, and introns. So the exons are predominantly where the mutations are. So these exons represent where the 2,000 mutations reside, or the most of them in that lead to CF. And each exon, which is combined together to make the mRNA, may have different and numerous mutations. Now I'm showing you an example of exon 12 here. And what you can see here is that each red bar represents a specific mutation. And the different mutations occur at different frequencies. So for example, the G542X mutation, which you see on the left, it has occurred in about 1,416 patients that we have in the CFF registry. In contrast, you look at a mutation like L558S, also in exon 12, but only 12 people in the registry have this particular mutation. So this sort of highlights the complicated nature of the different mutations and something we have to tackle with genetic therapies. So as I alluded to in my earlier slide, we could target the underlying cause by either improving protein function, and this is where the modulators have been a huge advance. We have two different types of modulators called either correctors or potentiators. The correctors work to improve cell protein movement to the cell surface, and potentiators work by improving chloride movement through the cell, and they're combined in Trikafta. And so by combining these different types of modulators, we get a lot of clinical benefit. But in contrast, we could actually go more upstream and target mRNA or DNA replacement or editing and lead to improved CFTR protein function. Now, it's important to remember that we need to target the lung. It is the lung that is the cause of most of the morbidity and mortality in CF, and we really need to make sure that the genetic therapies that we're developing are able to be delivered efficiently and work in the lung. So this is a table that sort of highlights in a very organized fashion what we're talking about. So we're talking about either mRNA, gene delivery, or gene editing. And potentially these therapies would be eligible, each person with CF would be eligible for each of these particular types of treatments. Now, the dosing schedule may be different for different therapies. For mRNA and gene delivery, it's likely that the treatment would have to be repeated for reasons I'll talk about in a second. For gene editing, it's unclear whether we would need to repeat the dosing or not. Now, in terms of where it be delivered, at least initially for mRNA and for gene delivery, we'd have to target the lung, which is important. For gene editing, it may be that we may be able to give the therapy systemically. Now, whether it is something is permanent or needs to be repeated for mRNA and gene delivery, it's likely that those would need to be repeated in an ongoing fashion. Gene editing, we don't know whether you could potentially do a one-time treatment, would need to be done on an ongoing repeated fashion. Now, when we talk about the modulators with rescuing protein versus our genetic therapies, it's important to consider the similarities and differences so we understand what we're dealing with. The similarities between modulators and genetic therapies, the first is that they both target the underlying cause, which is CFTR. They're both novel approaches, something we've not had up in the last few years. And they also have, in the case of modulators, had a long developmental timeline, and we suspect that that'll be the same case for genetic therapies as well. But what about the differences? Well, CFTR, as I mentioned, targets a protein. For our genetic therapies in development will target mRNA and or DNA. CFTR modulators are oral, taken as pill form. Nebulized therapies will be probably the first thing developed in terms of genetic therapies for mRNA and DNA. There could be down the road an intravenous formulation. In contrast to targeting the whole body with modulators, initially we'll be targeting the lungs with mRNA DNA treatments. And in terms of the studies, the way the studies will be done, in terms of the modulators, there were many patients who are eligible. And so we could have a sort of normal developmental pathway with respect to phase 1 studies, phase 2 studies, and phase 3 studies, and how we assess whether they worked and were safe or not. Because there's a relatively few number of people who will be potentially, at least initially, eligible for genetic therapies, we're going to have novel study designs and novel ways to assess whether something is working or not. I won't go into too much detail here, but this is something that we should be aware of as we develop these new therapies. So who are eligible for these genetic therapies? Well, clearly people who, based on their gene mutations, are not eligible for modulators are in need of additional treatment options. There are also people who may not tolerate modulators for side effects or other reasons. And so if they can't take those modulators, they need something to target the underlying problem. And there are people who have relatively modest clinical effect. Not everyone has a robust response to Trikafta, and people who have modest responses may be looking for additional treatment options as well. And finally, you can make an argument that every patient with CF has the need for potential treatment options, because Trikafta, as well as it works, is not curative at the end of the day. So an argument could be made that everyone has additional treatment options needed. What about the registry data in terms of who is not eligible for Trikafta? If you look at people over the age of 6 who are ineligible for Trikafta, what you see is there's a higher proportion of Black patients and Hispanic patients who are in need of additional treatment options because they are ineligible for Trikafta based on their genotype. So clearly a population of need, as I alluded to earlier. So now to transition about the specific therapies that we are going to be developing. The first thing I want to talk about is the mRNA therapy. The idea here is that regardless of your gene mutation, if we're able to deliver a normal mRNA into the airways, then that mRNA will be used to make normal CFTR protein, and which can carry out its normal function. One of the challenges of mRNA treatment is that it doesn't last for a very long time in a cell. So the protein will be made only for a limited amount of time. And so this will require the treatment to be re-administered in a regular, ongoing fashion. This does not require that the mRNA be delivered to the basal stem cells, but to the airway cells that normal require CFTR. With respect to gene therapy, again, this is the idea that we take a patient regardless of their gene mutation, but we deliver into their airways a normal copy of the CFTR gene, which is then makes a normal mRNA and then can make normal protein. So it would be eligible. Every patient would potentially be eligible. Again, as with mRNA, that the gene would eventually be lost over time, and so protein function would be diminished over time, and so redosing will be required. And as with mRNA, we do not require these gene therapy treatments to be delivered into basal stem cells. And finally, gene editing. This is the idea that you actually take the mutation and you correct it such that it now functions as a normal gene. And once it functions as a normal gene, the mRNA that's made is normal, and the protein will be made will be normal and carry out its normal function. Now, if an edited cell divides, such as a basal stem cell, then the new cells will contain the same edits. And so you could have a perpetual self-renewing population of an edited, corrected cell. However, if an edited cell dies, then the edits will be lost, and then you'll be unable to make the mRNA and protein in a normal fashion, and may need to be repeated. as I mentioned, the ideal editing target in this way would be the basal stem cell, which is a self-renewing population. So you'd be carrying on indefinitely. The technologies we have today are relatively inefficient for editing. And so one of the challenges going forward is to make programs that are very efficient with respect to editing. Another way to think about these genetic therapies is to think about the analogy of a car. In this way, let's think of a car as representing a CFTR protein. And CFTR mutations in this analogy represent car problems. So how can we solve that? Well, one way is to take a car that's not working and just rent a car. And we would have access to our car rental. We'd be able to do our normal day-to-day work and functioning. And this is in some ways represent what mRNA would be. Again, we would have a rental for a fixed period of time. We'd need to rent the car again. With respect to gene therapy, as opposed to a used car rental, we'd have a new car, which would belong to us. This would, again, be available for us to use on an ongoing fashion, but this would be available for a period of time, and then we would need to get a new car to have our normal functioning. And finally, the idea of editing is that as opposed to getting a rental or a new car, we actually repair the car that is malfunctioning and repair it in a way that it can function normally. We can carry out our normal day-to-day functioning. And this repaired car would belong to us. It would potentially be available for us for the indefinite period. And this would be DNA editing. Now, in addition to the different technologies, one of the things to keep in mind is some of the challenges that we have to overcome as we develop these genetic therapies. And one of the challenges is to be able to deliver the gene, whether it's mRNA or gene therapy, to the right cell. As it turns out that the airway is composed of different types of cells, and the CFTR protein is made in different proportions in these cells. Now, the basal stem cell is sort of the cell from which each of the other cells are produced. And the secretory cells, which is shown in the middle here, is a cell that makes most of the CFTR. So if we're thinking about delivering gene therapy or mRNA, we have to make sure that they get into the secretory cells in order to replicate what we'd expect to happen in a normal fashion. Ideally, if we get in the basal stem cells, then we would that would be a self sort of correcting in that if it's in the basal stem cells, it would also be produced in the secretory cells. But this is technically a difficult thing to do. In addition to getting into the right cell, we have to make sure that the genetic therapies get into the right portion of the lung. There's two big components of the lung called the conducting system, which is the airways, and the exchange surface, which is the alveolar space where the carbon dioxide and oxygen are exchanged between the lung and the blood. The conducting system is, however, where CFTR is most abundantly expressed. And this is where we have to get our gene therapies or mRNA to be taken into these cells at this particular part of the lung and make sure they're getting there in an efficient way and do the most benefit. So that's one of the challenges we're going to have to evaluate is whether the therapies we're giving to our patients is getting into the right portion of the lung, which is the conducting system. And finally, there are challenges specific to CF patients. One of the things we know is that CF mucus tends to be sticky, thick, and acidic, and it's going to be damaging to the genetic therapies, whether it's mRNA or gene therapy. So it may be difficult to get into the airway if there's inability to get through the mucus. There's also immune cells that can take up and destroy or neutralize the gene therapy. Lung cells, as we mentioned, are continually replaced. So this may require repeat dosing of mRNA and DNA replacement. And finally, basal stem cells, which we talked about, would be advantageous to get into, tend to sit at the bottom of the airway and hard to reach. So this is another challenge that we'll need to overcome. Finally, I just want to talk a little bit of the timelines that we may see the development of these therapies. If you look at the example of modulators, Kalydeco was the first approved modulator, which was applicable to 10% of folks. Then Orkambi, Symdeko, and finally with Trikafta, and we were able to get to a treatment which was potentially eligible for 90% of people with CF. And that took about 15 years from the first studies to Trikafta being approved. So a long development timeline. And the remaining 10% of people with CF who are now not eligible for Trikafta, I think we will take an incremental process of developing treatment that would potentially be applicable to every person with CF. So I think we are going to take a little bit of time to get to where we have treatment for every potential person. But this is something that's going to be an ongoing process, and we're all committed to. And finally, I just want to update you on what programs are coming to clinical trial in the next short period of time, in the next year or so. In terms of mRNA treatments, there are 3 programs by Arcturus, Recode, and Vertex Pharmaceuticals, which you may have heard of. All have mRNA programs they're ready to bring to clinical studies. In terms of CFTR gene delivery, 4D molecular Sparavant have programs that are about to begin very soon. And Crystal Biotech actually has started a program in Australia. So look for some results from there, hopefully the next year or so. And parenthetically, I just want to mention 2 other programs from Elox and Splicesense. Technically, they're mRNA programs, but their particular treatment would be applicable to people with specific mutations. And the idea behind their treatments is that you help your own cell make your own mRNA as opposed to giving exogenous mRNA. But this is something to be aware of. So with that, I'd like to thank you for your attention. Just highlight the idea that we are working very hard at this. This is something that's going to be incremental. We're going to learn more as we go forward, but this is something we're all committed to developing for our community. And with that, I'd like to turn it over to Dr. Lynn Cotel, who's going to talk to a little bit more detail about the studies that are coming down the road. Thank you for your attention.
Great. Thank you very much. Nima, thank you so much for sharing your story. It really is inspirational, and thank you for being so willing to talk about it. Manu just gave us a really great description about the science behind gene therapy trials. And what I would like to do in the next 10 minutes or so is to review what we are doing as a community to move these studies forward and to help people with CF and their families learn about these clinical trials. I have no disclosures, related to this study. We are all participants in clinical trials, and we don't always realize that we are doing that. We do that both in our personal lives and as patients. The collection of information provides opportunities to improve health outcomes for ourselves and also for our community. For example, as we heard in the opening keynote presentation, the CF Foundation Registry collects robust data from patients throughout the country, and there are similar registries in other countries. The registry is a powerful tool. It allows us to do research every day. Through patterns identified in the registry, we have learned about the relationship between lung function and nutrition that actually transformed clinical guidelines many years ago. Sputum samples are another great example of research that we do every single day. Collection of sputum cultures led to our understanding of how organisms such as Pseudomonas aeruginosa and B. cepacius spread among people with CF, and this led to the development of our infectious disease guidelines. There is a very specific life cycle to clinical trials. Phase 1, and when a drug is being studied, it starts with a phase 1 study, and then it ends with what's called a phase 4 study. Phase 1 studies are very small. They tend to run through very quickly, and they look primarily at safety. They're very short studies. Phase 2 studies are a bit larger and longer and help determine safety, efficacy, and what's the right dose to put on for the optimal drug. Phase 3 studies happen after the phase 2 study is done, and those are even larger, and they provide proof of safety, efficacy, and the exact dose needed for FDA approval so the drug can come on the market. How long it takes to complete a clinical trial varies, depending upon how long it takes to see the desired impact of the drug, but also on how long it takes to complete enrollment. But it doesn't end there. As we've heard several times today, and for those of you who listened to the Trikafta talk a little while ago, phase 4 studies look at what happens to an approved medication once it has been on the market. Clinical trials build on each other, and they direct new research. In our day-to-day life, cell phones are a really, really good example. In the early cell phones were very simple. All we could do would be to make a call and receive a call. Can you imagine that? Our phone calls today are mini sophisticated computers. You can track where your friends are at all times, and you can make plane reservations or hotel reservations. Modulators are another really good example. The first modulator, as we heard before, worked for a small percent of the population. But those other early studies provided the building blocks to develop modulators that now work for 90% of the CF population. We expect the same thing to happen with gene therapy. It will start by focusing on a small segment of the population. But as we get more experience, we expect they will expand and they will include more people. So what do we learn from clinical trials? Clinical trials do not always lead to new therapies, but they always lead to improved knowledge. Although, again, we typically think about clinical trials focusing on the development of new medications, we do research for many reasons. For example, we study how to improve clinical care through observational studies, again, using the CF Foundation Registry. Sometimes clinical trials actually teach us things about CF that we did not previously know. So, for example, years ago, we learned about the normal fluctuations in liver function that occur in people with CF through data that was collected during a clinical trial. But not all clinical trials are about drug discovery. We have studies that look at ways to support mental health using cognitive behavioral therapy. And again, as we've talked about a few times during these sessions, follow-up during phase 4 studies provides information about long-term safety and side effects. In the Trikafta study, there was a lot of discussion about some of these side effects. An example being mental health issues as a possible side effect of Trikafta. Another example is that early on, again, we learned that one modulator drug was not going to be enough, and we needed to add a second and then a third drug. So what's different about gene therapy this time and the gene therapy trials? As we just heard from Dr. Jane, we know that as a community, we've had lots of fits and starts with gene therapy. And you may really be asking, rightfully so, what's different? But it's really different, as we just heard. Science moves quickly. And over the past 2 decades, as we have seen in the previous presentation, we have new technologies which have successfully been used for people with other diseases. So we really are optimistic. Nonetheless, immediate success is not guaranteed. These studies are going to be complex. We have a small pool of patients who will initially be eligible. These studies will require long-term commitment and may in fact require travel. We recognize that the logistics of these studies are going to be challenging for everyone, but we're optimistic that, like modulators, each study will build upon the previous study to ultimately provide treatment for everyone. So what's in it for you? If you fall into the 10% of people who are not eligible for a modulator, or you do not tolerate modulators, these trials are targeted towards you. The goal is that ultimately this will be for everyone. But for now, you have the opportunity to participate in this new frontier and help the CF community move into a new era. Patient safety is the #1 priority for every clinical trial. Based on the studies for gene therapy that have been done to date, we do not anticipate significant safety issues, but we know and expect that this is a major concern for everybody who participates in a clinical trial. Your safety is protected on a multitude of levels throughout a clinical trial. I will briefly go through this graphic, and we will post it into the chat. It can also be found on the CF Foundation website under the Research tab. The take-home message here is that before a study is allowed to proceed, it is reviewed and approved by 4 separate groups, all of whom prioritize safety. This includes the FDA, the Therapeutics Development Network, the Data Safety Monitoring Board, and the Institutional Review Board of each study site. The Data Safety Monitoring Board specifically follows all of the data once a study starts. They meet on a regular basis to review the study and, as needed, should any safety concerns arise. During the life of a study, these same 4 groups routinely look at how the study is proceeding, primarily in terms of safety. The TDN and the Data Safety Monitoring Board have patient representation, so the voice of the patient is always heard. As a patient in a clinical trial, you are in the driver's seat. It is your decision to participate, and it is always your decision to continue. Your team's job is to help you make the best decision for you and your family. Over the next few minutes, I will review what we have been doing as a community to move this work forward. A multifaceted gene therapy working group comprised of scientists, clinical care team members, and members of Community Voice was convened last year. An educational subgroup of the gene therapy working group was charged with developing materials about gene therapy for patients and research teams. We recognize that everybody needs to be on the same page. This slide demonstrates the timeline of the educational subgroup. We started with focus groups to hear from people with CF what they knew about gene therapy and how we could help them learn more. Over the last few months, we have developed more educational materials for people with CF and research teams so that we can communicate effectively. This will now be expanded to clinical care teams. This will continue through the next year and beyond. Our goal is to ensure that everybody has the tools that they need to move forward. These resources will enable people with CF to make the best decisions about clinical trials that feel comfortable for them and their families. These include webinars, meetings such as today's, handouts, updates to the CF Foundation website, news stories, videos, and social media blasts. And many of these materials will be available in Spanish. So how can you be part of the process? As I mentioned in my opening slide, we do research every day, and it is through research, whether it is observational, basic science, or drug discovery, that we will continue to improve the lives of people with CF. So how can you be part of that? Ask your care team about research studies. You can look up what trials are being done on cff.org or clintrials.gov. As new studies open up, these lists will be updated. Learn about gene therapy. We will be sending out a new glossary explaining all the terms. Trust me, it's confusing. We all need to learn new words. You will also see a handout with sample questions to ask your care team about gene therapy. We want to make this so that you have the tools that you need to learn about this. Learn about your mutation. Understanding the many CF mutations is an ongoing project. There are still a lot of mutations that we are learning about. And if you don't know about your mutations or their significance is not clear to you, talk to your care team about getting them tested again. We are learning more all the time. Most important, though, please share your voice. Join Community Voice. We are all in this together. Thank you so much.
Thank you so much, Dr. Jane and Dr. Quattel. The information presented was so informative, and it is so refreshing to hear that I can help play a part in research that will be something for everyone. Now, we have a few questions that we've received from the ResearchCon registration webpage that I'd love to ask you. For those who are attending the session today, please remember to type your question in the Q&A box, and we could get your question answered. Or you could get your question answered first. As I mentioned during my presentation, modulators don't work for everyone, especially people of color and certain ethnic groups. Will genetic therapies include these groups in the future?
Absolutely. So as we heard in Dr. Jane's talk, and as we've heard, I think, in some of the other presentations, there are about 10% of CF people who are not eligible for modulators or who do not tolerate them. And within those groups, there's a higher percentage in people of color, people of Hispanic descent, and some other subgroups of certain ethnic groups that are not eligible for modulators. These studies are really ideally for those people, and those people will be eligible for those studies really from the get-go. But again, our hope is that eventually it's going to be available to everybody.
Great. Our last question is, who will be involved in the clinical trials you mentioned? Will these start with a certain age demographic? Will the trials be able to look at transplant recipients?
Right. So traditionally, children are often excluded in the initial segments of clinical trials. Children are considered to be especially vulnerable, in part because they actually can't give true informed consent for themselves. So these initial studies will not include children, they will include adults. And as we heard from Dr. Jane's talk, the initial studies are really going to target the lungs. So for these beginning studies, transplant recipients will not be eligible. But again, our hope is that over time, everybody's going to be eligible.
Great. This is incredible information. Thank you again so much to you, Dr. Quattel, and to Dr. Jane. I'd also like to thank Dr. Clancy, who has been answering questions in the Q&A box. This has been so inspirational and provides so much hope for those living with CF. Now I'd like to quickly pass it back to Shelby and Cindy George to close us out.
Oh my goodness. Thank you so much, Nima and Dr. Quattel and Dr. Jane, who just gave the biggest and quickest, deepest dive into everything. And I'm very sorry that we haven't been able to answer all the questions that we need to. So keep an eye on the Q&A to get a reply, hopefully from Dr. Clancy. And Nima, it's so important how much all of our stories help further the much needed research that we still need to do. And I cannot believe that we are at the end of the conference already this year. It's gone by so fast. Please remember that there are many wonderful resources related to this discussion and the resources document found on the reception page. Please spend some time exploring the resources, download anything that you want to keep, and the ResearchCon environment will be open for the next 15 minutes to allow you a bit more time to explore. Unfortunately, it's just a tight time restraint this year, and the event will end after that.
Any sessions that you have missed are now available under the replay button on the right-hand side of your screen. You can come back to this site using the same link that you used to access the event and watch any sessions that you may have missed. Only the English version is available right now. In a few weeks, the Spanish version will be available as well on the Foundation YouTube channel. And you'll receive an email notification when all these are available.
Thank you all for attending ResearchCon this year. This has been an absolute dream come true for me and for Cindy both. We hope that you will complete the post-event survey, which you will receive via email immediately following the event. We greatly appreciate all of your feedback, good and bad, so that we can prepare better for next year. And we hope that we were able to live up to everything that we tried to give.
No, thank you, Shelby. And thank you again for joining us today and the other day. And we look forward to seeing you at other community conferences throughout the year or back here at ResearchCon 2023.
See you soon, guys. Let's connect offline.